nobody reacted the way you'd expect. In July 2024, a paper was released based on data from NASA's Perseverance rover. Not a press conference, not a dramatic announcement, just a document. Dense, careful, almost restrained to the point of discomfort. Inside it was a rock, not visually impressive, about the size of your hand, collected from a place called Jezero Crater, a dry basin that, billions of years ago, held a lake. The kind of environment where, on Earth, life doesn't just appear, it leaves marks. Perseverance drilled into it, sealed the sample, and ran it through a suite of instruments. Sherlock, Pixel, Supercam. Each one designed to answer a question we've been asking for decades in slightly different ways. Was Mars ever alive? What they found wasn't an answer. It was a pattern. Organic molecules, not unexpected on their own. We've seen those before, even in meteorites. But here, they were arranged in a way that made people pause. Carbon-based compounds, clustered in veins within the rock, associated with minerals that typically form in the presence of water. Water that wasn't passing through quickly. Water that stayed. And then there were the shapes. Tiny structures, just tens of microns across, smaller than a human hair, embedded in the rock matrix. On Earth, formations like these often trace back to microbial activity. Not proof, but a suggestion with a direction. That's where the argument starts. Because if you followed Mars exploration at all, you know this pattern. Something looks like a biosignature, and then the room splits in half. One side leans forward, cautiously, but you can feel it, saying this is consistent with microbial life, ancient, simple, long gone but not erased. The other side leans back. Abiotic processes can do this too. Chemistry is more creative than we like to admit. Given enough time, pressure, and the right environment, non-living systems can produce structures that look uncomfortably alive. Both sides are correct. That's the problem. And the gap between them isn't small. It's not a missing data point you can fill with one more experiment. It's structural. The kind of uncertainty that doesn't shrink easily. Because we're not looking at life. We're looking at traces of something that may have existed nearly 4 billion years ago, in a place that has spent most of that time being sandblasted by radiation, stripped of atmosphere, and chemically altered beyond recognition. Imagine trying to reconstruct a sentence after burning the book, scattering the ashes, and waiting a billion years. That's the level of signal we're working with. And yet, the reason this matters isn't Mars. It's what happens here, on Earth, if the answer tilts even slightly in one direction. If those structures are biological, even tentatively, it means life didn't happen once. It happened at least twice, independently, within the same solar system. That shifts the probability landscape in a way that's hard to overstate. Suddenly, the galaxy doesn't feel empty in the same way. But if they're not biological, if this is chemistry mimicking life so convincingly that our best instruments can't easily tell the difference, then we have a different kind of problem. It means we might already be looking at alien worlds and misreading them. Or worse, we might never be able to say with confidence what we're seeing. There's a line in the paper, easy to miss if you're skimming, that basically says, these features are consistent with, but not diagnostic of, biological activity. That sentence is doing a lot of work. Consistent with life, not proof of life. And that gap between consistent and confirmed is where scientists are still arguing, months later. Not loudly, not in headlines, but in journals, in conference rooms, in carefully worded rebuttals. Because this isn't just a scientific disagreement, it's a boundary problem. How do you prove life existed somewhere when all you have are fragments that can be explained without it? And more importantly, what level of certainty are we willing to accept before we say it out loud? There's a quiet tension running through this entire situation. NASA is already preparing the Mars Sample Return mission, a joint effort with ESA to bring these sealed tubes back to Earth sometime in the early 2030s. That's when the real analysis happens, with instruments too large to send across space. But even that isn't a guarantee. Because if the ambiguity we're seeing now survives that level of scrutiny, then the question doesn't go away. It becomes permanent. And there's something almost unsettling about that. We've spent decades assuming this is a question with a clean answer. Yes or no, life or no life, signal or noise. But Perseverance may have stumbled into something more complicated. A gray zone, where the evidence never fully resolves and the argument becomes part of the discovery itself. And that brings us back to the rock, the one collected in Jezero crater. Because where it was found, and what surrounded it, might matter more than the sample itself. We'll come back to that. The rock didn't exist in isolation. That's the first thing that gets lost when people focus too narrowly on the sample itself. Perseverance didn't just pick up something random off the Martian surface. It was navigating a landscape that had already been mapped, debated, and, in a quiet way, argued over for years before the rover ever landed. Jezero crater wasn't chosen because it was interesting. It was chosen because it was specific. Orbital data from instruments like CRISM on the Mars Reconnaissance Orbiter had already identified mineral signatures: clays, carbonates, the kind that form when water interacts with rock over extended periods. Not a flash flood, not a one-off event, sustained presence. On Earth, that's where you look for fossils. So Perseverance was sent there with a kind of quiet expectation. Not hope exactly, more like a probability distribution that leaned in one direction. And then it started moving. Over the course of 2021 to 2024, it traversed what used to be a river delta, layers of sediment deposited over time as water flowed into the crater lake. If you've ever seen a delta from above, you know the pattern: branching channels, slow-moving water, fine particles settling out in thin, repeating layers. That's not just geology, that's a recording system. Each layer holds a moment. Each mineral, a condition. Temperature, chemistry, duration, all of it gets written down in ways that are incredibly difficult to fake perfectly. Now here's where it gets interesting. The specific sample that triggered this debate, informally referred to in some internal discussions as part of the Chayava Falls sequence, wasn't taken from just anywhere in the delta. It came from a zone where multiple conditions overlapped: carbonate-rich rocks, evidence of past water flow, and critically, signs of chemical gradients. That last one matters more than it sounds. Life, at its most basic level, exploits imbalance. It feeds on gradients, differences in energy, in chemistry, in environment. On Earth, microbial life often clusters exactly where these gradients exist, where oxidized and reduced materials meet, where water moves slowly enough to allow interaction, but not so fast that it wipes everything clean. So when Perseverance found organic molecules embedded in carbonate veins, in a place where chemical gradients likely existed billions of years ago, you start to see why some scientists leaned forward. It's not one signal, it's the alignment of several. But alignment isn't confirmation, because here's the counter-argument, and it's not weak. Mars is a chemically active planet even without life. Ultraviolet radiation breaks apart molecules in the atmosphere, creating reactive compounds that settle onto the surface. Volcanic activity in Mars's past introduced heat and pressure conditions that can reorganize minerals in complex ways. Even water, by itself, can drive surprisingly intricate chemistry. There are lab experiments, some going back decades, where purely inorganic systems produce structures that look, under a microscope, disturbingly biological: filaments, tubes, clusters that resemble colonies. Not alive, but close enough to make you hesitate. So when critics look at the Perseverance data, they don't see life being hidden, they see chemistry being underestimated. And there's a deeper issue here that doesn't get discussed enough. Our entire framework for identifying life is based on Earth, not just loosely, completely. We know what life looks like because we evolved alongside it. We know the signatures it tends to leave: certain isotopic ratios, specific molecular structures, patterns of organization. But all of that is derived from a single example: one. Which means when we go to Mars and look for life, we're not really asking, is there life here? We're asking, is there something here that looks enough like Earth life for us to recognize it? That's a narrower question than it seems, and it creates a bias that cuts both ways. We might see lifelike patterns and assume biology when it's not there. Or we might miss something genuinely alive because it doesn't fit the template we expect. There's a quiet irony in that. We built incredibly advanced instruments, Sherlock mapping organic compounds with ultraviolet fluorescence, Pixel analyzing elemental composition at microscopic scales. And yet the limiting factor isn't the hardware, it's interpretation. At one point, a researcher involved in Mars sample analysis compared the situation to trying to identify a language from a handful of burned pages, without knowing what language family it belongs to. You might recognize patterns. You might even guess correctly. But you can't prove it without context. And context is exactly what's incomplete here. Which brings us back to something I mentioned earlier. The idea that this might not resolve cleanly, even when the samples return to Earth. Because better instruments don't automatically eliminate ambiguity, they just push it to a finer scale. You might identify more complex organic molecules. You might map isotopic ratios with higher precision. You might even find microstructures that look more convincingly biological. But unless there's a definitive biosignature, something that cannot be explained by any known abiotic process, the argument doesn't end. It sharpens. And that's already starting to happen. Some teams are focusing on the spatial distribution of the organics, not just their presence, but how they're arranged relative to the mineral matrix. Others are looking at the geological history of Jezero, trying to reconstruct whether the environmental conditions truly supported long-term habitability. And then there's a smaller group, less visible, but increasingly relevant, asking a more uncomfortable question. What if we're expecting the wrong kind of evidence entirely? There's one detail from the rover's journey that keeps resurfacing in these discussions. Not the rock itself, but what sits just beyond it in the stratigraphic sequence. Layers that suggest a transition. From a wetter, potentially habitable environment to something far more hostile. A shift. And if you trace that shift carefully, it starts to hint at a timeline. Not just of water appearing and disappearing, but of conditions changing in a way that might have allowed something to emerge and then erased most of it. That's where the conversation gets quieter. Because now you're not just asking whether life existed, you're asking how long it had, and whether what perseverance found is a snapshot of something that barely had time to begin, or something that lasted long enough to leave a deeper mark, one we just haven't recognized yet. We'll need to look at that transition more closely. That transition layer, the one just beyond the sample, is where the tone of this whole story shifts. Up to this point, you can hold two positions comfortably. Either Mars had the right conditions and maybe life emerged, or it didn't, and what we're seeing is chemistry doing a very convincing impression. But when you start mapping the timeline of Jezero crater more precisely, the argument becomes less about if and more about when, and for how long. Because the lake that once filled Jezero didn't exist indefinitely. Based on crater dating and sediment analysis, we're looking at a window measured in tens of millions of years. That sounds like a long time, until you remember Earth formed over 4.5 billion years ago. Life here may have taken a few hundred million years to appear, possibly less, depending on how you interpret the earliest isotopic evidence from places like Greenland's ʻOumuamua formation. So, Mars had water. Mars had chemistry. Mars had energy gradients. The missing variable isn't obvious, and yet, something didn't carry through. At some point, Mars began losing its atmosphere. Not suddenly, this wasn't a cinematic collapse, but gradually, over hundreds of millions of years. The planet's magnetic field weakened, solar wind stripped away gases, and surface pressure dropped. Water became unstable. Lakes evaporated. Rivers stopped flowing. Whatever habitable pockets existed began to shrink, then fragment, then disappear. If life ever started, it was suddenly on a clock it didn't control. Here's where the data from Perseverance becomes more than just a curiosity. The mineral context around those organic compounds suggests formation in relatively calm, stable water. Not a chaotic flood, not a short-lived event. That implies persistence. Conditions that didn't just flicker on and off, but held steady long enough for complex chemistry to organize itself. That's the part that leans toward biology. But the surrounding layers, especially those showing increased oxidation and signs of environmental stress, tell a different story. They point to a system under pressure, a transition from stable to hostile. And this is where a subtle reversal starts to take shape. Because for years, the working assumption was, if Mars ever had life, we'd find its strongest evidence in the oldest, most stable environment.